Carbohydrates are typically assumed to come from plants. Grains, legumes, fruits, vegetables, and root vegetables constitute the conventional list of carbohydrate sources in dietary education. The Greenlandic Inuit diet, operating in an environment where plant carbohydrates were seasonally limited or entirely absent, challenges this assumption in a way that has interesting implications for understanding human carbohydrate metabolism and the minimum carbohydrate requirements for healthy function. Glycogen, the stored form of glucose in muscle and liver tissue, is a carbohydrate. When an animal is killed and its muscle tissue is consumed fresh, that glycogen is present and available as a dietary carbohydrate source. The actual quantity varies depending on the animal's recent activity level, how quickly the meat is consumed after death, and how it is processed. An animal killed after intense exertion will have partially depleted its glycogen stores. Fresh meat consumed within hours of slaughter retains more glycogen than aged, stored, or frozen meat, from which glycogen is progressively converted to lactic acid during post-mortem glycolysis. The Inuit also derived carbohydrates from fermented animal products. The fermentation process converts some protein to carbohydrates through microbial activity, and the stomach contents of herbivorous animals like caribou contain partially digested plant matter, including glycogen and other carbohydrates. These sources are irregular and modest compared to an agricultural diet, but they are not zero, and the traditional Inuit were not in a state of absolute carbohydrate deprivation. The broader metabolic point this topic opens is the question of minimum carbohydrate requirements. The dietary reference intake for carbohydrates, 130 grams per day, is set based on the brain's estimated glucose requirement. But this recommendation assumes glucose as the brain's only fuel, which is not accurate. During adaptation to very low carbohydrate intake, the liver produces ketone bodies from fatty acid metabolism, specifically beta-hydroxybutyrate and acetoacetate, that the brain can and does use as an alternative fuel with high efficiency. Many individuals on ketogenic diets report stable cognitive function at carbohydrate intakes of 20 to 50 grams per day, well below the DRI. The caution here is directed at the tendency to treat the ketogenic or very low carbohydrate approach as categorically superior for all people and all purposes. The Inuit adapted to their dietary pattern over many generations, including, as research has revealed, through genetic variants affecting fatty acid metabolism that are more prevalent in Inuit populations than in others. These variants influence how efficiently the Inuit body processes and utilizes polyunsaturated fats. A person without those adaptations moving to a very low carbohydrate diet may not experience identical metabolic outcomes. Individual variation in carbohydrate tolerance is substantial and real, and the appropriate carbohydrate intake for any individual requires attention to personal response, not only to what an ancestral population achieved.